Hi, this is Edward Awad. In this last video on cellular respiration and fermentation, we will look first at fermentation, then I will give an overview of how catabolic and anabolic pathways are interconnected, as well as how cells regulate their metabolic activities. If you recall from previous videos, we saw that glycolysis is a common pathway for both cellular respiration and fermentation. In the presence of molecular oxygen, cellular respiration is favored, but when oxygen supply is low or insufficient, some cells can divert pyruvate metabolism to the fermentation pathway. So let's look at what happens inside the cell when oxygen is in low supply. If you recall, complex 4 in the mitochondrial electron transport chain is responsible for transferring electrons that originate from electron carriers to the final electron acceptor, which is molecular oxygen. When complex 4 does that, it is reoxidized and therefore can accept more electrons. But when there is an insufficient supply of oxygen, a cell cannot reoxidize complex 4, as the electrons can go nowhere. So complex 4 remains in its reduced form. This causes the entire respiratory chain to become reduced. NAD+ and FAD are not generated from their reduced form, and pyruvate oxidation stops due to lack of NAD+. Likewise, the citric acid cycle stops, and consequently, ATP production stops. And if the cell has no other way to obtain energy, it dies. Luckily, some cells under anaerobic conditions, meaning in the absence of oxygen, continue glycolysis and therefore maintain the production of ATP, but in limited amounts. They are capable of doing that through fermentation, which is a metabolic pathway that evolved for the purpose of regenerating NAD+ from NADH to keep glycolysis going. Two fermentation pathways are known to exist: lactic acid fermentation, which results in the formation of lactic acid as end product, and alcohol fermentation, which produces ethanol as end product. In lactic acid fermentation, the NADH molecules produced in glycolysis are used by the enzyme lactate dehydrogenase to reduce pyruvate. This redox reaction uses the reduction power of NADH to convert pyruvate into lactate, and in this process replenishing NAD+. Lactic acid fermentation occurs in some bacteria and in muscle cells when they are starved for oxygen. Alcohol fermentation involves the use of two enzymes to metabolize pyruvate. First, pyruvate is decarboxylated to produce acetaldehyde. This results in the formation of carbon dioxide. This reaction is catalyzed by the enzyme pyruvate decarboxylase. Then acetaldehyde is reduced by NADH, replenishing NAD+, and producing ethanol. This second reaction is catalyzed by the enzyme alcohol dehydrogenase. Alcohol fermentation occurs in yeast cells. The main purpose of glycolysis and cellular respiration is to produce ATP to meet the energy needs of cells. A secondary purpose of these pathways is to produce intermediate metabolites that are used as substrates for the synthesis of lipids, amino acids, nucleic acids, and other biological molecules. It is through these intermediates that different metabolic pathways in cells are interconnected. Looking at catabolic interconnections, here are some examples. Polysaccharides are hydrolyzed into glucose, which passes on to glycolysis. For lipids, they are converted to fatty acids and glycerol. Fatty acids are in turn catabolized into acetate. The acetate is converted into acetyl-CoA and fed into the citric acid cycle, while the glycerol part is converted into an intermediate in glycolysis. Proteins are hydrolyzed into amino acids, which would feed into glycolysis. Let's look now at anabolic interconnections. Gluconeogenesis is a process by which amino acids are metabolized into intermediates of glycolysis and the citric acid cycle, which are then used to form glucose. Acetyl-CoA and intermediates of glycolysis are used as precursors for the formation of fatty acids. The citric acid cycle intermediate, alpha-ketoglutarate, is the starting point for the synthesis of amino acids as well as purines. Oxaloacetate, which is another intermediate of the citric acid cycle, is a starting point for pyrimidines. Cells regulate their interconnected metabolic pathways depending on their needs and in order to maintain metabolic homeostasis. They can do so by regulating the activities of key allosteric enzymes through feedback inhibition or feedback stimulation. Let's take the example of these hypothetical pathways. The end product C of the first metabolic pathway is the starting point of two different metabolic pathways, one that is catabolic, leading to product G, and the other one is anabolic, leading to product E. If cellular conditions result in the accumulation of product G of the catabolic pathway, this product acts as an allosteric inhibitor of a key enzyme, let's call it enzyme 1. This inhibition results in the accumulation of product C. At the same time, product G acts as an allosteric activator of a key enzyme in the anabolic pathway, let's call it enzyme 2. The accumulation of product C and the activation of enzyme 2 result in a condition where the anabolic pathway is favored over the catabolic pathway. This switching between metabolic pathways is analogous to intersecting railroad tracks with enzymes acting as switches. Let's take the example of the allosteric control of glycolysis. The main control point in this metabolic pathway is the enzyme phosphofructokinase, which catalyzes the phosphorylation of fructose phosphate into fructose bisphosphate. This enzyme is allosterically inhibited by excess ATP and citrate and activated by excess ADP or adenosine diphosphate and AMP or adenosine monophosphate. In the citric acid cycle, the main control point is the enzyme isocitrate dehydrogenase, which converts isocitrate to alpha-ketoglutarate. NADH and ATP are inhibitors of this enzyme, while NAD+ and ADP act as activators. In addition, citrate acts as an activator of a key enzyme that commits acetyl-CoA to the synthesis of fatty acids.